And today we're going to pick up from where we started last time, which last time we covered PCA, and we talked a little bit about how to load our data for PCA, how to define different groups. We talked about how to explore our data from different perspectives. And let's talk today a little bit more into what to do with this analysis. How does it actually help us understand what are the different properties of the data set that we're dealing with? And we'll continue talking about what's called unsupervised machine learning. So before we go into that, let me briefly kind of give you an overview about what is machine learning. So most of you probably have heard already about machine learning. It's a machine that learns, right? And we have these two types of machine learning methods. The first one, the logic behind it, is when we ask some kind of an automated process about patterns. So find patterns, show me what do these patterns reveal, right? It's asking the machine to identify some pattern. And that's called unsupervised machine learning. Why is this an important part of the process? Just like we saw in exploratory data analysis, these patterns help us identify technical issues. They can help us spot outliers. They can help us identify correlation or similarity between different samples. They're really there to help us spot patterns that using statistical methods, it's difficult to find. The second option is when we want to train a machine. So we want to create a machine that will go on and do something for us. So it's just automating a process. For example, if I know what something looks like, but I don't want to just sit there and keep on looking for it, I want a machine to look for it. And that's called classification or supervised analysis. And that supervised analysis is based on training. So we want to train some kind of an algorithm. Now, that's not necessarily a good approach all the time. So we should really reserve that for when we are familiar with the data, we've prepared the data, we have a high confidence that our data is representative of the classes that we have. And so a lot that has to do with machine learning requires both biological interpretation, validation, and kind of a critical review before we actually trust those results. There's a great video where you ask a person to organize shapes, but only give them a few different options: a circle, a square, or a triangle. Now, if you come across a shape that looks like a star, you don't have a place to put it. So you will still put it into some kind of a shape that it fits into, for example, a circle. So it's very easy to imagine how classification or machine learning can make a mistake, have a bias. So unsupervised machine learning is what we'll talk about today. The basic idea is that we have some source data. We'll look at that source data. We apply some kind of an algorithm. Typically, it's going to be dimensionality reduction or clustering, because these methods look at distances between all our variables and describe those distances in terms of similarity. So whatever is closer, right? So distance could be measured. So now we can identify things that are closer together, farther apart. And so the result, the new information from such machine learning would be the group. So based on that distance, if we do principal component analysis, does not tell us the answer, what groups we have, it just shows us what we have. But clustering will actually assign each sample to any particular group, and that is new information that we have. So principal component analysis, remember that it looks for variance, and that variance means that we can identify some kind of a plane in a multidimensional space. We can find what are the dimensions of our data in that multidimensional space. And then we can look for ways to maximize variance so that our projection of the real data is the most representative of this variance. And that's really what these principal components are. Principal component one would be the most highest variance. Principal component 2 would be the perpendicular. And so we end up with this projection, essentially, that helps us understand the variance between samples based on all of the data that we have. Now, similar to this, there are several other types of ways to measure distance. For example, PCA looks at variance. NMDS, which is uh, the non-metric multidimensional scaling, is to collapse information from multiple dimensions, just like that, just like we have, but it uses primarily Euclidean distances in principal component analysis, and NMDS uses rank orders. And so that is typically applied to data that is not continuous. For example, if we look at metagenomic data, which measures abundance of species grouped by similarity of sequences, we get data that has gaps in it. So it's not fully continuous, it has gaps, and so it's more like ranks rather than continuous data. Another way to look at variance between samples is to create a new measure, which here you can see t-SNE. So this measures measures distance in something that's called perplexity. So t-SNE stands for t-distributed stochastic neighbor embedding, which is a technique that is very well suited, but also not so easy to use for high dimensionality. So the more genes we have, the more variance we have in the data that we have for the genes. For example, in single cell data, there's a lot of zeros. It's called zero inflated data. And for genes, they're not standardized, right? So what is perplexity? Perplexity is a measure for information that is defined as 2 to the power of the Shannon entropy. And the perplexity of a data with k sides is equal to k. So in t-SNE, we're really optimizing some additional parameters to maximize variance between groups that are more similar to each other. Now, this leads us to the question, if we have an ability to measure distances, could we use that distance metric to now assign samples into groups based on separation, right? So what if we have separation? We assume that separation exists. Could we measure some kind of a number of groups? So a good approach to this is hierarchical clustering. Here we have some samples. Let's imagine they are in a principal component analysis dimensionality space. And we can say, take the closest samples and assign them to be one cluster. So these two are going to become one. These two are going to become one. These two are now farther apart. So they're the next level to being farther apart. Then after we've done that, we can combine them further into groups of whatever's closest. Up until when does the algorithm stop? The algorithm stops when everything is in one cluster. So if we do this procedure, let's try to see what it comes out to be. Hierarchical clustering will give us a hierarchy from the bottom, which only 2 are grouped into 1, and the top, when everything is grouped into one single cluster. So let's take a look at how to do that. So I'm assuming that most of you have started Transcriptomics 3. I've seen that Lydia has been making progress with that. Others, if you have not started yet, we highly recommend looking at it because it will really give you a better understanding of what these methods do. So let's take a look at server.tbio.info. So please let me know once you are here and logged in. Okay, so maybe let me know if you can log in and you're there, and we'll go through several examples here for clustering. Harpreet, could you please paste a link here to the 15 gene data set?
Okay, Leah.
Okay, thank you, Fatima. and that's okay. If you can move forward with some of that transcriptomics coursework, that will help you get caught up. Okay, anyone else? Did anyone else make it to the server? All right, thank you, Rudina. Yes, thank you. And I'll show you also where from this place you can get some R scripts that later on we will modify to apply the same methods in R. Okay, so here we have supervised analysis and unsupervised analysis. So when we talk about clustering, is it supervised or unsupervised?
Supervised. Unsupervised.
Unsupervised, right? So it's unsupervised because we're not telling the clustering method to find something for us. We're asking, what do you see in the data, right? So let's go to unsupervised analysis. Okay, we're waiting here for Harpreet to give us some data, but I will just show you one time with the data set that I have on my computer. And you probably have this data from previous exercises that we did. Thank you. So here we have a number of data sets. Let's take a look, for example, on this 15 gene data set. Okay, so we have data set that, for example, has groups in it, right? And so you can find, here's a data set that has the IDs of our samples, it has group names, and then it has 15 genes. So Harpreet, this is the one that is marked, right? You have the one that is with groups. Harpreet?
So we need a data with the group, right? Okay.
Yes.
Okay.
Okay, so make sure that your data has groups in it. And what I'll first do is I'll run PCA in R. And remember that even in R, we specify several things, just like we did last time. For example, scaling. Do we want to scale our data? Do we want to center our data? And what does transpose do? Who remembers what does transposing data do? Why do we transpose data? Does anyone remember?
We transpose. Sorry.
You have two things open. Yeah, go ahead.
We transpose data to change columns and rows.
Right. So if we want to use PCA to look at genes, then we don't have to transpose. If we want to look at samples, then we have to transpose, right? So here it's already on yes. Let's click on save, and we will get our PCA 15 genes marked, right? So exactly what we did last time in R. Here we are going to run this so that you can try looking at the result, okay? And now, based on what we did with PCA, can I do it slowly? Yep. Yep. All right, so file upload. Here I add files, and I will take the file. Remember that it has groups, so it's called 15 genes marked. Okay, Harpreet, you pasted a link to 15 gene transposed. We don't need the transposed. We just need 15 genes marked. Please put that in there. Okay, again, for anyone kind of exploring the data before you look at it, you have the IDs of your samples as column names, and then you have a separate row that has the column group name, and then you have all of your data. Okay, so we choose this for upload, and it is uploaded. Then I click on Start, PCA-R library. Here I don't change anything. And then here I click on End. Okay, so how many of you were able to run this pipeline? Harpreet, do we have the data?
I'm just finding the link for that data set specifically.
Okay, here, let me find the link. Okay, this is the link right here. You see I have ID group. So this is the data right here. All right. So let's take a look. I have the previous one that I did. And let's take a look at the output. And please still let me know when you have run the pipeline. Just put in the chat, I'll see it. So what are some of the outputs here? The outputs here include the PCA table, because as you remember in R, what we did was we produced an object called PCA res, right? It was the result of PCA. What it is essentially a table. So what does this table look like? This table shows us the projections. So it actually prepares a table, and the table has, instead of genes that define each sample, has principal components that define the table. So for example, I have principal component 1, principal component 2. Remember, they show us the most variance. How do I create a chart from these two components? What kind of a visualization would be the proper one to use to visualize such data?
The logarithmic transformation.
What kind of?
The log transformation.
No.
It is already in logarithmic transformation.
No, it doesn't have anything to do with logarithmic transformation. These are just numbers, right? So I have numbers here. What would be a good way to visualize this data, right? Right now it's a table. How would I visualize it? What kind of, should I use box plots? Should I use a bar plot? Should I use a scatter plot? Should I use a heat map? What would be a good way to visualize this data?
I think we could run different plots. The first one you mean?
Okay, so the correct answer is scatterplot. Yes, thank you, Rudina. Scatterplot, because we have two continuous sources of data. This is continuous, right? This is continuous. You can see it goes from about 0 to whatever, right? So if I create here a scatter plot, what will it look like? Does this look similar to what we got in our practice last time? Yes. All right. So because why? We see 3 groups, right? So these 3 groups correspond to how many actual groups that we know from our clinical data. Okay, so whenever we had, let's take a look at our input data. We have our input data that already has some groups, right? These are clinical groups. These are subtypes of breast cancer. So if we want to understand how does PCA correspond to what we know about our data, okay, we can take this information and we can insert our data right here, right? So here I now have this information. Okay, let me undo this. Place it right here. I'll call this group. So if I want to understand how does this data correspond to groups, all I need to do is I need to sort this. And now I can make sure that I select the data appropriately right here. So my first group is going to be basal. And I can take here and say, this is my x-axis, this is my y-axis for all of my basal types. My next one is going to be cloud in low. Again, I take the x-axis and I take the y-axis. Then I take my luminal group, x-axis, and y-axis. And finally, I have the fourth group, which is normal-like. And for normal-like, this is x-axis, and this is y-axis. Okay, does this make sense? Does anybody have any questions about what I'm doing? Okay, so no questions, right? Now, tell me, what is the group that is most different from everything else? Which group here, and maybe this is not very big, so let me make this bigger. Which group here? Okay. Yes, this is now a two-dimensional plot, right? This is one dimension, this is second dimension. Okay, Fatima is saying cloud and low. So why cloud and low? So cloud and low is this one, right? The orange ones are cloud and low. These are the luminal ones. So in terms of distance, okay, Rudina is saying luminal. In terms of distance, where is the distance here? How do I measure distance? Is this closer, or is this closer, or are these closer? How do I know? The answer is this distance is represented by percentages right here, because these percentages show us variance. So in terms of these numbers are not on the same scale. Even though here I have -10 and +15, here I have -10 and 6, this axis actually represents a bigger distance because this represents 53.5% variance between my samples overall. So every distance on the x-axis is approximately 2.5 times bigger than distance on this axis. So here, the distance, the correct answer is luminal. Luminal is the most dissimilar from the rest. And then within the second group, that is the second biggest distance, is this cloud and low from the rest. Does that make sense? All right. So now we saw one of the outputs, right? And that was this table. Now, thankfully, we have a plot here that already makes this chart for us. And we can see, what does this plot produce? So this plot shows us the same thing, right? It shows us how we have our principal component 2 is the y-axis, and it's about 18%. Principal component 1 is the x-axis, about 53.5%. So the distance between this group, which is luminal, and this group, which is cloud and low, to the rest, right? We know that this distance is bigger. Why is it bigger? Because the variance is represented by percentages. So if you multiply percentage by the actual distance, you will get a bigger distance between this group and the rest, as opposed to between this one and the rest over here. Okay, Rudina, does that make sense?
When you say the distance, for example, the luminal group, if you could see this, could imagine the center, it's very close to basal. So you're thinking that, can you see that has one very close to the basal group, one outliner?
Okay, so now you're bringing a good point, right? So the point is, how do you actually measure distance? Where do we measure distance from? Do we measure it from the edge?
Imagine the center?
Well, yeah, but this actually brings us to our next point in the discussion. So let's think about this a little bit more when we do our next exercise. So here, the point is that this distance is bigger than this distance. But you are correctly asking the right question. But when we say distance between this group and these groups, what do we take as our point where we start from? Is it the center? Is it the border, which would be right here, right? Or the center? Or is this some kind of an outlier that somehow could be eliminated, right? So there are many ways to measure distance. You are absolutely correct. So let's now think about this, because right now we are just visualizing our data, right? We're just looking at it with our eyes, and we're just kind of, measuring kind of what we see. But in real life, we are only seeing one of the projections. And here we have multiple options. For example, this is another projection. We have PC1 and PC3. This is the same data, a different projection. Here's another projection, PC2 and PC3, right? And the projection is completely different. So we have many projections that we could potentially consider. And the question is, could we go through all of the options with our eyes and measure distances and then conclude some kind of a conclusive answer? What are the actual groups that we have and how to divide them best? Not what we know about the clinical data, but what we actually have in the data. And this is the job for unsupervised analysis like clustering. Okay, so let's ask that question, and we'll use hierarchical clustering to ask that question. So let's now go back to unsupervised analysis. And in unsupervised analysis, we don't have that option to take data that has groups in it. So if we want to take groups and forget about the names of our samples, we just need to replace the names of our samples with the group names. But we won't do that because we already have our, we have a table that tells us what it is. So let's just take this example, which is cell lines 15 genes. It won't have the groups. Okay, to give you this data, I'm going to give you the data real quick here. Okay, so 15 genes, not this one, this one. Okay, so please, what happened? Okay, so please take this data set. And now we will upload this data right here. Okay, make sure that you've got the right data. Continue. And then instead of PCA, we will look at clustering. And the first example, we will use H-cluster, which stands for hierarchical clustering. And now we can define what kinds of distances we're actually going to use. Okay, so what are the distances? Euclidean is the same distance measure like we use in principal component analysis. Okay, so let's keep it at Euclidean. Linkage means exactly what you were asking, right? If I have two samples that are close together, how do I make them into one? So if I look over here and I say that here are two dots that are very close together, how do I link them? Okay, that's linkage. And so you can have multiple ways of linking, and we will use this method called Ward D2. So the Ward D2, it's named after a person whose name was Ward, and he created this hierarchical clustering approach that basically says that two closest samples to each other are going to be linked into one, and the center of that new cluster is going to be right in the middle of them, and that's called Ward D2. Do we need to transpose our data? All right, Rudina says yes, we'll do it. How many clusters? When do we need to transpose our data? So we need to transpose our data based on what we want to measure. If we want to measure distances between genes, so again, look at our table. What are the columns here? What are the column names here, Fatima?
Let me see the column names. Are those
Right
types
here.
of genes?
No, types of genes are always ENSG.
Okay.
So types of genes are rows. And what are these names?
I'm not, well, the top row says their IDs in the
They're
corner.
sample IDs. Yeah, they're sample
Okay.
IDs, which these are names of cell lines, right? So these cell lines were derived from patients. All of these patients had breast cancer of different types. And those cell lines could be grown in the lab. So we could try different drugs on those cell lines, for example, right? Which you can't try on patients, but you can do it on cell lines. So here, these names are cell line names or sample names. So when we want to ask the question, do we have a question about samples or a question about genes?
In this case, we have questions about the genes, right?
No, about the samples, right? It's just
Okay.
that. So these dots right here on PCA, these are samples, right? We're saying that Claudin low, what's Claudin low? Claudin low is a type of a breast cancer. It's not type of a gene.
Okay.
Okay. So we are, and Rudina, thank you. Yes, we're asking a question about samples. So because we're asking the question about samples, we need to transpose, because naturally all of these algorithms will use your names of rows as the objects. So these, in machine learning, these are called objects and features. Features are genes, objects are samples. So we do transpose. True.
Okay.
Now,
Thank you so
how many
much. That
clusters?
provided a lot of clarity.
Okay, so why don't you answer the next question? How many clusters do you think you expect? So based on
4.
4. Perfect, let's put 4. Why don't I try 4? Some of you try 5. Some of you try 3, right? Because what if this is one and this is another one and this is another one? Okay, how many have started the pipeline? Okay, Ludmila, have you started the pipeline? Rudina, and Sylvia? Or should I repeat this? Okay, thank you, Rudina. Lydia and Sylvia? Okay, so Lydia, let's try to get you unlost. Where would you like us? Do you want to go through this again? Let's try this again. Okay, so Sylvia and Lydia, could you tell me when you're in this section, unsupervised analysis? So click right here and tell me when you see what I see on my screen. Okay, so you're there. Okay, Sylvia, are you here?
I understand, Professor. I am observing your class.
Okay. All right, well, just to make sure that you can do it, you know, we can wait for you. But okay, so Lydia, if you're here, let's upload here the file. You have the file, right, Lydia? Okay, so let's upload the file together. So here we have the file. It doesn't have any groups. Choose for upload. Okay, 100%. Continue. Press on start and H cluster right here. So hierarchical clustering. Now, what is hierarchical clustering? As I showed, it takes the same samples in our multidimensional space and assigns the closest ones to be one sample. It's like a pseudo sample or a cluster. And it goes from the bottom, so all of our dots, the closest ones, then if that's considered to be one dot, the next closest one and the next closest one, up until it gets to the top. So you have all of your clusters in a single cluster. Okay, now here you have several parameters. Ward D2 is the one that we're using for linkage. Distance stays Euclidean. Here you can choose 3, 4, or 5. Why 3, 4, or 5? Because if you look at this PCA, you can say this is 1, this is 2, this is 3. That's 3 clusters that I expect. Or you can say here's 1, here's another one, here's another one, here's another one. That's 4 clusters. Or you can say 5. Maybe this is a cluster on its own. This is another one, this is another one, this is another, and that's another one, right? So you can choose any number that you would like that's reasonable to find within this PCA. Okay, Lydia, does that make sense? Where are the output files? Yeah, okay, well, we didn't get there yet, right? Here we transpose, save, and then end. So you say you already ran the pipeline, great. Let's take a look at the results. So here is my pipeline, and here are some output files. So I have several different results. One result is the PDF, right? So always PDF is going to be some kind of a visual. So let's take a look at that PDF. Did you get the PDF, Lydia? Did everybody get the PDF?
Yeah, I got the PDF, but I did one modification. Instead of using the one of the parameters as the Ward D2, I use the average.
Okay, that's fine. So we'll see what the difference is. But there shouldn't be a lot of difference because in this case, we only have 15 genes. It's actually not super difficult, but let's see what we see from this result. What does this result show us? So this result shows us that we have two major groups, right? So this we see right here. So if we cut off at the top height, we kind of look at from the very major trends that we have in our data, we have this group and we have this group. If we go a little bit lower, we see 1, 2, and 3, right? 3 groups. And if we go even lower right here, we have 1, 2, 3, and 4. If we go lower, we have 1, 2, 3, 4, and 5, etc. Right? So the height is a distance that we can measure, and we can cut it off to get a certain number of clusters. Now here, it's not very easy to see which one belongs where, but you can right away spot different things. For example, you can see that this guy right here, HCC 202, is an outlier. Or you can see that all of these samples form this type group right here. These form this type group right here. These form this type group, right? So you can kind of start seeing some structure to the data. But since I asked the specific question, what are the samples and which groups do they belong to? Let me open up this result right here. So again, what I took was the full table result right here. Okay, let's take a look at this full table. And I have here object. Remember, objects are my samples. And then I have a cluster ID. Okay, so now these objects are in the same order like what I have in my input data. So if I go to my input data, not here, because remember here I reorganized them. If I go to my input table, which was right here, right? So this is the same order. We can check 184A1, 184B5, right? It's the same order. So I can now take this group information that I have right here, or just take it from here. In fact, I'll take it. Yeah. So I'm going to take it from here. I'll say group. And I'll just copy this over to here. So now I can again organize it by my clinical data, what I know to be true about my data from the clinical perspective, and check whether the clustering did a good job. So I should see the same cluster ID like in my group definition. So here did a correct job, right? All of these are number 2. All of these are number 4. All of these are number 3. All of these are number 1. Does that make sense? So the machine, this algorithm, the clustering algorithm, I didn't tell it what the groups are, but it automatically found them for me. So if it automatically found them for me, that means that I can trust it to find patterns, okay? So now the question is, how does it actually do this? What is the process by which it can go through the sample data that I have with the 15 genes and the 52 samples, which is a lot of data? If I was to go through this manually, it would be very difficult for me to spot these patterns. How does it actually do this? So another output that I have here is the R package, okay? So to understand how this does, how this works, we're going to take a look at the R package that will produce the same output that we did here, but we'll also take a look at what does this correspond to in terms of the patterns in gene expression. So we're going to look at the expression data, right, which is right here. You can see that this is our actual expression data, and this is the output from clustering. So I'm going to pass it on to Harpreet, but just before I do that, I will do one last thing here. The way we describe these numbers is by saying some numbers are high, right? So for example, this is a low number, 0. This is a pretty high number, it's 6. What if I mark these, right? So I can go in here and do conditional formatting, and I can say highlight cells, color scales, and let's say the high would be green, the low would be red. Okay, this will help me see the patterns a little bit better. So since I organized my data already by group, can I see some patterns that are specific to each group? For example, could I see something that is specific to basal? Okay, so I have here a clear pattern, right? Red, 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 red. If I go to cloud and low, I again see some specific pattern, right? Here I have all of this is red, but this is unique, right? It doesn't appear in any other group. So this is some kind of a pattern. And then all of these. So we can see here that these patterns actually define what the clustering does. Now, if I'm just looking at these patterns, which ones are closer together? Do I see some that are closer to each other? Which ones do you think are closer to each other? Okay, cluster number 3 is close to what? Median. Would you say that maybe normal-like and basal are kind of similar to each other because all of this is pretty high and this is also fairly high? And then I have these markings, right? So this is, or low, sorry, this is low, this is low, this is low, this is low, this is low. And here I also kind of have a similar pattern, right? So for example, basal and normal-like seem to be fairly close to each other. So if I look on my PCA, I can kind of see that, right? Basal and normal-like are closer to each other. You see that? So this is actually what the distance represents, this kind of similarity. All right, so Harpreet, I'll pass it on to you and maybe you can explain how to do this in R so that we can see some of these outputs and heat map.
Thank you, Ilya. Okay, so once you are on there, so just type your answer yes, so that we can perform this hierarchical clustering in R. So just type yes once you are here. Okay, Sylvia, that's great. So we have this 6th lesson that is dedicated for this clustering. So apart from Sylvia, who is able to log into this OmicsLogic platform?
Um, uh, excuse me, I am just listening. I would prefer to pay attention because,
Okay.
please, thank you.
Okay. So does anyone able to log into this OmicsLogic platform? And if you are there, okay, so I think we can practice it if you're not able to. I think this lesson might be free for you, but if you need access, okay, so we can actually, I mean, you can later look into that. We can look into that, that it should be the,
The number 6 clustering code.
okay. So if you are not able to, so let's, I mean, I can show you how we can actually perform. The first of all, so let's take the same, I mean, the same data set, only I have a this transpose data here. So we can actually just go one by one step. So the first part, again, explain it. If you go to the next part, so in the code breakdown, you will learn exactly which function doing what. And then once you're able to complete that, the next you will find one exercise here. So if you run and able to run this code, so as you are, okay, so you will Bipsa is going to mail you this code.
Yes, Dr. Harpreet, you can continue.
Okay, so let me just share the link of this file first. The data set that we are using, the same data set. So only we have a transpose data here. So here we have ID, sample ID in the first column. Then in the second column, again, we have these group names, and then followed by the genes that we have in this data, that is 15 genes. So first the libraries that we need. I think you can open any of your, okay, so like if you have access to any lesson, so you just clear that lesson and just console, clear the console, and let's do this clustering step by step. So first we need two libraries. So the libraries that we need, that is stats and tools. So first we need to load these library, these packages. So then we are loading this data. So I'm creating an object where we are going to store our data. So with read.table, then we have to provide the path of this file, input data. Then we have to define whether our file contain header or not. So header equal to true. Then we will define separator. So why we need to define separator? That here if how our columns in our data are separated from each other. So if you have CSV file, then you define comma. If you have tab separated file, then you define separator as tab. The next step, if your data, if your header has like the same, last time even Lydia has this question, why we define check dot names equal to false. So this command we use whenever if you are in your header there is same name of the sample, something is same, so that those samples should be remain as it is. They should not be changed into 1 2 3 or something like that. So we used to define that. Then it is string as factor. So as in our data we have string, like here if you look at the second group, so the in the second column we have these strings, so that these strings should be taken as string in our data. It read it like that, so that's why we define string as factor. Then we used to define strings as factors, sorry, equal to true. Then row names, if your input data has already have rows, so you want to define that these are the row names, so then for that we used to write row dot names equal to 1. So we actually, Lydia, we giving a command so that our name should not be changed. So that's why we keeping it as false. So then, if does anybody remember how we can check our whether our data get loaded or not? So with which command we can check whether our data get loaded or not? Dimension. So dim command, we can check how many rows and the columns are there. So we can do that. Dimension is equal to data. So here we have 52 rows and 16 columns. So out of, because in one column we have group. So the another then 15 genes are there. So total 16, and then we have already defined rows and header. So once you have that, the next before doing a clustering, let's see how we can plot a heat map. So for visual, if you look at now this data, head is equal to data. So here we have gene names and then genes, and then we have a groups. So next is we want to plot heat map for that. For that, first we need to remove the second column which contain groups. So how we can do that? So let's define it as df. So the data, then we're going to take from the second column to the 16th column. So here we are extracting only numeric values, and those numeric values are we have transferring those into an object that is df. So then to draw a heat map, we need only numeric values, and for the heat map, it requires it as a matrix. So then next we will convert this data as matrix. So as with command as dot matrix, we are transferring a data frame into a matrix. Then with heat map, we can just define and create our heat map. So here we have, then we can define, as if you look at this here in our rows. So in our column, we are unable to, it is not clearly visible, although these are the genes. So we can define the size 0.5 with sex dot call. So it becomes very small. We can actually change it. So does any one of you able to produce this heat map? That's great. So then this is only with this, here we are defining the size of the text. If you look at this, so you can even transpose this file. So if you want to look at, you want that your column should, your rows should be genes or your sample should be in the column. So what you can do, you can actually transpose this matrix. So like here we have mat. So what we can do, we can do a simple command, that is T, and then we convert this matrix into the transposed matrix. So now you can take this matrix, that is mat underscore T. So sex dot call is actually defining the size of your column. Here, the size of this column legend, it is defined that. So like here, we can actually increase it as well. If you look at this now, these are your columns, right? Here in the heat map, these are your columns, and here these are your rows. If you want to change or you want to adjust the size of these columns, so these are our samples and these are our genes. So with six dot call, actually we defining the size of column, and if you want to change the size of this text, that is on the rows, so with the six dot row, sorry, R row, you can define its size 0.8. So this is how you can just create your heat map and look at its expression label, each of these genes in all these samples. Is it clear to everyone?
Excuse me, Harpreet. Now I understand that in this case, in these examples, the rows are the samples and the columns are the genes.
So I have just transposed it. So you can actually take it original as well. Like here, first I have, suppose I just want to take original. So what I can do, I can take it as mat. So I have, which is not transposed. Here I have transposed that with this T command. So if I take its original matrix, so now you can draw this plot again like this. Now your in the column you have a genes, while in the row you have samples. So you can transpose, if you want to transpose, then you can use this command. Is it clear now? Sylvia, is it clear to you now? Okay. Oh, sorry, Radina. Okay, so this is how you can actually look at your data and you plot it, and then you can actually define different color as well. So right now, now let's proceed with how we can do a clustering. So once we have this data, that is DF. So we have this data here. The next is how we can create a hierarchical clustering. So here, what we need to do, that first we need to provide our input data here. So here what data that we have with the hclust, this is a method. So with hclust method, this is a method with which we can actually create hierarchical clustering. So with this method, we can create our hierarchical clustering. Then we need to provide, we have to create distance matrix. So then what for to create a distance matrix, we need to define methods. So you can actually choose different methods. Already Ilya has shown you that we have Euclidean method. So you have another methods as well. Then you have to define method for the linkage. So like you can, here you can define complete, or whether you want to define single or average. So you can choose those method accordingly. So this with this, and then with the hclust, we are creating this object. So this is how you got the same cluster dendrogram that you got there. So here you can change these methods as well. So let me just paste it here. First, we need to define which method we are using, like hierarchical clustering, hclust. I'm just pasting its link here. So does any one of you actually able to log into this particular lesson, or this lesson is accessible to you or not? Whether this lesson is accessible to you now? Okay, so this is how we can actually, if you are still not, this is not accessible, so the code must be provided to you. You can access that lesson later as well. So here we are just, I have just pasted this command in the chat box. Here what we are doing with the hclust, so we are providing the method that we want to create hierarchical clustering, and then we are creating a distance. This is for the distance, we're creating actually distance matrix. Then we are taking our input data, that is df. Then we are defining the method. Here you can play with these methods that you know that different methods are available. So you can play with those methods, like for the which kind of distance metric method you needed and which kind of linkage you needed, right? Complete, average, or single, or W3D. So you can actually play with that. Then once and create an object and provide this hierarchical clustering object into the cluster object, then just plot. So with the plot, you will able to produce this hierarchical clustering, this clusterodendrogram. So does any one of you able to produce this dendrogram? Okay, Rudina. Apart from Rudina, anyone else who is able to produce this dendrogram? Or if not, okay, Lydia. Okay, so this is how we can create this dendrogram. Okay, Sylvia. So does anyone have any issue till now? So I think already Beepsa has shared the code with you. If you use that code, all the lessons in this course will be freely available to you if you use that code. Beepsa has mailed you that code. So these are
Dr. Harpreet,
Mhm.
just to answer and address Lydia's questions here, Lydia, there are a few free courses on OmicsLogic, while the rest of them are premium. The premium access comes at $10. For all program participants, please refer to all the session emails as well as the invites that I sent you. There are oftentimes more information than just the Zoom link or the video link. Please refer to the first emails from session 1 and the welcome emails where the code and how to utilize it is given. Please go through the following steps so that you're able to access these courses and you are ready for the session. So if you're not able to follow the session right now, you can again go back, watch the video, and do it once you have access to the full lesson. Thank you so much. Thank you, Dr. Harpreet.
Okay.
Harpreet, a question, please.
Mhm.
When is this course going to finish? At the end of February?
I think the last date that we have, the last session that we have on, just let me check.
It's 12th of February is the last session.
Thank you. And your platform is going to be available after the course is finished?
The platform itself is going to be available for anybody who has an advanced license. It would be available for them for an extra 30 days. And the others will have their access until the 14th of February. And if you're especially going to join any kind of like a project, so that would be up for discussion.
Okay, thank you.
Yeah. So, and for anybody who have an advanced license and is going to be working, so you will have more than February 12th, which is like your access will end on March 12th. So you will have that time to plan your project, work on your project, as well as have the one-on-one sessions with your mentor. So that is it. Yeah.
Excuse me,
Mhm.
to have the advanced course, we will require to subscribe again.
No. You do not have to subscribe again. I will let you know about this if you're interested. So please let me know. If you already
Yes,
have an advanced license, just let me know and I will be able to take it beyond the session. Okay.
yes, I would like to be registered to the advanced section. I require the platform available
Sure, Sylvia. So I
more
will reach
time.
out to you after the session. Okay. Now let us
Okay,
continue the session.
thank you
Yeah.
very much.
You're welcome.
So next you can actually play with different, you can use different methods. For example, you have already learned this is average. We have average method. So instead, oh, sorry. So this is complete method. Then you can actually play with these methods and look at how these different plots, you can generate a different plots and look at the differences between them, right? So like here you just define, if you define average, sorry, we have to plot this as well. Cluster 1, I'm just creating with the average. Then we need to plot this. So you can actually compare after applying different methods. Suppose this is already, well-known data. So then you can actually play with these methods and these parameters so that you will able to obtain the best dendrogram for your own data. So for example, here, if we looking at this method, it almost produce similar kind of plot. But if you change another, apart from the average method, then if you define another setting, so like you want to, you instead of the Euclidean method, you are defining centroid method. So if you, how your results are changing with that, you can analyze and then pick up the best result accordingly. So this is where you can actually define the different method. So for example, let me just copy the different methods that we have. For example, we have Euclidean, Manhattan, Canberra, then binary. So similarly, for the linkage, we have centroid bar D2. For example, here we defining var D2. And then we are defining the same Euclidean method. I'm just copying this same. So I'm changing the third method, that is bar D2. Bar dot D2.
I have
So
one
then we
question.
can, yeah.
For the distance, the all logarithm, it just the is the
No, not Euclidean. We have Manhattan, we have maximum, then we have different methods. So you can play with those methods for the distance as well as this for the linkage.
right. Okay, I understand
Yeah.
that. Okay,
So let
the-
me just paste this. I mean, the different methods that are available. You can explore this as well in this stat library. So let me just paste the different distance metric methods that even more than that are available, but I'm just let me paste it here. So for example, for the distance metric, we have Euclidean, Manhattan, maximum, Canberra, then binary. So you can play with these methods and then look into which method is actually representing or clustering your samples in a best way, right? Then for the linkage, we have these single, complete, average, bar D, bar D2. So then you can actually plot for each of these methods and then look at which representing the best. So for example, here I am doing this cluster plot. So I think you can explore these methods, and you might have once you will get access to these lessons, and even not the lessons, I think there is not much that we require. We need to define just hclust method, then we need to define the distance metric method and method for the linkage. This is how we can create, and then we need once we have created this object for this hierarchical clustering, then for that object we need to write a plot, then you can plot that. So if you have any issue or anything that still you are not able to understand, you can ask me right now in this code if we have done, and then we have actually can write and all this uh, objects and the results of the hierarchical clustering in form of a table as well into the file.
Thank you.
Okay. Is it clear to everyone? So this is how we can play with the we can write this plot, and then if you are interested, like you want to do analysis with and you want to store this information into the table. So for that, what we are doing, we just need to define with this simple function cut dot tree. Here once you have this subject, then you need to define only with this cut tree, and then you can even define the number of the classes that you want, and then you store this these id into the file and write into a file. So if we have already this plot, then we need to define only
A question, Harpreet. May I use R to evaluate nucleotides?
Sorry?
Yes, we can use nucleotides.
Nucleotides. So are you asking for the sequence data?
Yes, the sequence data for R or machine learning. Is it possible to use the same code?
So it depends. I mean, if you have sequence data, so usually if you have a nucleotide or mostly protein, nucleotide sequence. For that, I think there are a number of methods that convert them first into the features. They create the features based on nucleotide composition. Then they have like 5' composition, 3' composition. So based on the composition, then they actually create one numeric data for those nucleotides to find the differences, and then you can do these clustering and all on numeric data.
Okay, thank you.
Yeah, because for any sequencing data, so for the sequencing data, eventually this kind of all analysis you can perform mainly on your numeric values. So to create the numeric values, usually the number of features are computed for those nucleotide sequences, right? So if you like, you are interested in, you have some sequence belong to some particular species, but to then from that sequence, then uh, the features are created. So there are a number of the methods that actually create features either for the protein sequence or nucleotide sequences.
I would like to use R for this, for the evaluation of data sequences. It will be possible?
So it depends upon, are you are looking for like SNP or something, exactly what you are looking for. I mean, this is depending upon, suppose if you are like trying to classify the groups based on nucleotide sequences, it is not directly possible. If you are like, you want to apply this machine learning, but you have to create uh, you have to create features, that is numeric features. So there are a number of methods for that. But if you are like looking for mutation data, right?
Yes, exactly.
So for the mutation data, actually you have the approach is different, not is just like we are proceeding like this. So then you have to like, you if you are looking, I think, have you gone through the genomics lesson?
Not the lessons, not yet.
Courses,
I
I
am,
mean, if you are interested in some nucleotide sequencing data, I mean, how we can analyze and how we can find out the mutations in a sequence. So then there is this genomic lesson, introduction to the genomics, as well as the genomics one. So you can actually, you have most probably you all have the access to all these lessons, right? So if I'm looking at here, so these are the courses.
mhm.
So in the introduction to the genomics, and the genomics one actually deals with that kind of data.
Oh, okay. Mhm. And uses R also?
So then for the R, we have actually, yes, we are dealing, but we are not, if I'm even at this, let me just go to that lesson as well. So if we go to the courses, I don't know whether you have access to that lesson or not, but there is a genomics lesson. We have how we can actually read a sequence files. So here we have these, mainly these two lessons, DNA structure variants and DNA replication. So this lesson, two lessons are also available in introduction to the bioinformatics course. Here, getting started with the bioinformatics. If you are there, you will find here these lesson 3 and 4 actually deals with that. If you have that access, I don't know, because these are premium lessons. So actually here you are learning how you can read DNA sequence data in R, and then how you can generate DNA replication, reverse complements, and all those things. And exactly you are also looking for if there is mutation in your data. But the main courses that
Excellent.
you actually, apart from that, here, if you go to this introduction to the genomics and the genomics one lesson, right? So under these lesson, actually you have what are different tools are available, and how you can analyze that, and how you can generate like phylogenetic tree and all those things. So, and what pipeline you need to apply on a particular data or particular type of analysis. So you can actually take these lessons and go through these lessons first, because they actually deals with your sequencing data.
Okay, thank you, Harper.
Yeah. Okay, so these are, if you want interested in transcriptomics or RNA expression data, the transcriptomics lessons. But if you are interested, like you have a sequence data, right? DNA nucleotide sequence. So for that, that is specifically mutational analysis. So these two lessons are actually for that only. They are dedicated to that, those kind of things. Okay, so thank you everyone. If
Thank
you
you.
have Okay, so even if still you have any issue, or once you able to access to this lesson and you can practice it yourself, and then you can play with this data, and even in your data, I mean, you can play with that. And if you face any issue, you can email us. So thank you everyone for joining today's session.